Good morning, friends and dear student. Welcome back into the TYBSc Mammalian Histology paper number 2. Here, today we will start with the discussion of liver. That is a part 3.5 from your syllabus. 3.5 glands associated with digestive system. Among the other glands, today we will discuss only a liver and the sagittal section of a liver. So here in front of you, this is a photograph of a liver, a mammalian liver, or more of you can say precisely it's a man liver. Generally, a human liver is a two bilobe liver, and uh, it is a all substance of this liver is made up of a single single liver cell. It's a single unit of a single functional unit of a liver is hepatocytes, or called say you can say it's a liver. That each hepatocytes is a large cells with a round open face nucleus with prominent nucleoli. And if you see, these hepatic cells are arranged in a small lobules, that is called as a hepatic lobule, and these hepatic lobules are a little bit irregularly hexagonal in shape. Liver externally uh, covered with a layer of a connective tissue that is called as a Gleason's capsules. They are polygonal and they form the connective tissue lining around the liver, that is called as a septum transversum. This liver produces bile as a excretory organ, and it plays role in a digestion assimilation of a digested food material. This liver as a substance is dividable into a large number of lobules. Each lobules consists of numerous small lobules. That exocrine secretion of this liver cells is called as a bile, and that bile is poured out from the liver cells into the very delicate bile canaliculi that are present in intimate relationships with the cells. Each liver cell, that is each hepatocytes, are connected with the bile canaliculi. From this canaliculi, bile drains into progressively large ducts that end into the bile duct, and that bile duct then connected with this duodenum to pour its bile secretion, and that helps in digestion of a fat when that reach into the duodenum. Now here, so diagrammatic presentation of a liver. We'll see some major parts, tubes entering and leaving that liver. So the location wise, liver is situated just below to this diaphragm, and all these grayish color portion indicate the liver parenchyma. Liver parenchyma means a singular unit of that liver is a hepatocytes, and that many hepatocytes come together to form the hepatic lobules, and such hepatic lobules come together to form this parenchyma or a liver as a substance. Okay, now here from this liver, excretion, that is a bile as a excretion or a secretory product of a liver, comes out through these tubes. These tubes are called as a common bile duct, which one branch of which goes into the gallbladder where bile is stored temporarily through these cystic ducts, or one other branch of this bile duct open directly into this duodenum part of intestine. Okay, now there is a branch of aorta artery which enter into the liver, and another portal vein is there which collect digested food material from the intestine and directly bring that digested food material into the liver, so that digested food material will come in contact with the hepatocytes, and there is a further digestion and assimilation take place in the liver. So here artery entering into the liver, this portal vein entering into the liver, and this excretory duct coming out through the liver. So when we will see that detailed hepatocyte and hepatic lobules, that time these three tubes play important role in arrangement or studying the structure of the hepatic lobule. So we are discussing these tubules here. Now here another diagram, you can see these liver lobules are there, and gallbladder, and that common bile duct is open into the this duodenal C-shaped structure of duodenum along with the pancreatic duct to pour the bile secretion for the fat digestion. Now, functions of liver. If we discuss this function of liver at this moment, that will help us necessity of different cell type present in a liver. While studying the histology of liver, we'll come across different types of cell and arrangement, and why that arrangement is such way present in case of a liver, that for that purpose we need to learn these liver functions right now. First thing is a liver act as a storage part of a emergency water. So it is a storage of a water. Liver stores vitamin A and vitamin D. Liver stores ions. Liver is a second reservoir of a blood. That means lot of blood is there in a liver. Liver act as a filter apparatus for blood. Now erythropoiesis, a breakdown of RBCs, is a major function of this liver. All that RBCs are broken down into the liver, and separated bile is then collected and poured into the duodenum. Then in a liver, RBC are worn out, that is depositions of dead RBC take place in liver. Liver is the most important center for formation of a limb. Okay. Thereafter, this liver is a hemopoietic in embryonic stage. In infants, you will find that development of RBCs take place in case of liver, the place of liver. Liver is excretory as it eliminates bile pigments. Excretion. RBC phagocytized by Kupffer cells and waste product that bilirubin secreted into the bile. Then fibrinogen, an important protein for blood clotting, is formed in the liver. A synthesis of different protein take place in a liver. Then prothrombin, important for the blood clotting, is also formed in the liver. Then alcohol metabolism takes place in the liver. Whatever ethyl alcohol we drink, that is broken down and detoxified in a liver. Carbohydrate metabolism takes place in a liver. Now here majority of carbohydrate metabolism it take place in a liver only. That glycogenesis and glyconeogenesis take place in liver. Liver regulates salt and water balance. Okay, it regulates salt and water balance. It is an important center for heat productions. During the winter, our liver works and it produces temperature. Liver also stored fat. It is a site for storage of lipid. So fatty liver, we must have heard this word. So that fatty liver, that means more excessive fat is stored into the liver. Deamination take place in a liver. Breakdown of amino acid take place in a liver. Then ornithine cycle completed in liver. One of urea product, that ornithine cycle completed in liver. Detoxification take place in liver. All that medicine and everything get degraded into the liver. lipogenolysis take place in liver. Many of these lipids are break down into the liver. Plasma proteins are formed in the liver. Albumin, globulin, these all are synthesized in the liver. Inorganic salts and coppers are also stored in liver. In higher animals, in mammals, you will find urea is formed in the liver. Lipogenesis takes place in liver. So many more functions are done in a liver, and the major site of these all functions have synthesis and excretions and detoxification site in a liver is a hepatocytes. That is a location where it takes place. That is role of, not actually it's a role of a liver, but at cellular level if you think it is a role of a hepatocyte. Then glycogenesis take place in liver, glycogenolysis take place in liver, gluconeogenesis take place in liver, and glycogenogenesis also take place in the liver. So majority carbohydrate metabolism processes take place in a liver. And so all we'll see one by one what cell types are involved in our different functions here. Now here it is a liver lobule is shown. Now liver lobule, this is a irregular shape, hexagonal shape structure that is called as a hepatic lobule or a liver lobule. Now this a single hexagonal shape structure, hepatic lobule, are made up of rows of hepatocytes. So all these smaller hepatocytes come together to form one lobules, and each lobule is then separated with the connective tissue membrane, and that's why it looks like a hexagonal arrangement. Now in three-dimensional you will see that these layers of these hepatocytes are arranged on disc-like fashion. Okay, so it is arranged in a disc-like fashion, and centrally they have opening, and all these hepatocytes are radiating towards this central canal, central centrolobular vein. That here it is called as a CV, that is centrolobular vein. Uh, Centro, that means middle of a lobule. So centrolobular vein it is called, and all that hepatocytes are directed towards, so it gives a hexagonal radiating shape to this lobule. Now while all these hepatocytes are going towards the centrolobular vein, they forms a gap in between. That gaps are called as a sinusoids. Okay, thereafter there is a portal triad thing is there at the junction of these hexagonal hepatic lobules here, where there two to three different sides of a hepatic lobules joins together. That locations they form opening of a three tubules vessels. That three opening of three tubules is called as a portal triad. Okay, this, so you will find this portal triad located on each corner of this hexagon. Now what that portal triad is, it is a it is a bunch, three blood vessel bunch in the form of that hepatic portal vein, hepatic artery, and bile duct. These three tubules come together in a location that is called as a portal triad. Now here you can see it very clearly. So this central opening is a central vein. This is a central vein, and all these structures directing towards the center, going towards the centers. It's directed towards the centers. These all structures are the hepatic cells, and the gaps between the hepatic cells that forming the hepatic sinusoids. Sinusoid is generally a word where it represent a gap between the cells. So here the gap between the hepatocytes is called as a hepatic sinusoids here. Okay. So and this portal triad thing, what we learned in our previous slide is portal triad is a union of three tubules, is a bile duct, and hepatic artery and hepatic veins. So the concept is that all uh, food material absorbed from the digestive system is collected through this portal vein and poured over these plates of hepatic lobules. So this blood with digested food material that pass through these different hepatic sinusoids and reach into the central vein. While it passes over these hepatic sinusoids and hepatocytes, when it come in contact with the hepatic sinusoids and hepatocytes, all the liver function take place. What we said, that detoxification, storage, then metabolism, glycogenesis, carbohydrate, everything take place when that blood with all nutrients absorbed from the system come in contact with the hepatocyte. All that liver function has performed, and slowly that um, converted blood, that means if there is a absorbed carbohydrate is there, that glucose either may be converted into glycogen, or if it is there is glycogen or it's amino acids, they are break down and new proteins are synthesized and everything happens, and that final product is poured into the central vein. And simultaneously this hepatic artery also pour blood, so there take place the exchange of that material and everything get poured into the central vein. But at the during the course, whatever the RBC and some material get break down and that gives a bile, these biles get absorbed into the hepatocytes and they pour that bile into the another side of sinusoids, and there another side of a sinusoid there is a blood bile canaliculi is present. Through this bile canaliculi, that all bile is secreted or flowed in opposite directions and poured into the bile duct or ductus terminalis. So here only the biles flow in against to the central vein, and all that blood flows towards the central vein. Okay, so here it's a color photograph of a section of a liver. Here we have to identify the central vein. So this is a central vein, and this boundary indicate the irregularly hexagonal shape of a hepatic lobules. So to identify that slide as a liver section, we must see these irregular shape hexagonal structures and a central vein. If you find that central vein, you try to trace at the corner where if you can see the triad there. So here we will see here, we can see here that is bile canaliculi and artery and vein. So to this we can refer as a portal triad as a central vein, and all these are the hepatocytes. Now here you can see the small white color cracks in between this mass or matrix. That cracks are hepatic sinusoids. Okay. Now here for simplification, how exactly this tube system works in association with the hepatocytes, so you can get clear in this diagram. Now see the same thing here, it is a central vein. These are all hepatic lobules. Okay, so this is the central vein, these are the hepatic lobules, and these are the portal triad. Now this portal vein pour blood through these gaps, and that blood flows towards the central vein. Here again the artery also pour blood in these gaps or sinusoids, and it flows towards the central vein. But you can see this green color structure here. Now every this